So thank you for joining us this evening. Tonight we're joined by Matthew, who's going to talk to us about cancer mutations. But first, a little bit about our housekeeping. So if you've joined us for the first time, this might be new to you. But our events are aimed to everyone to explore the science of genomics and what it means to all of our lives. So we respectfully ask everyone participating, I know you might be curious and want to jump in with questions, but if during Matthew's talk, if you can put in your questions in the chat and I'll be moderating them, or if you can save all your questions for the end, then you can either unmute your microphone or put it in the chat or anything like that. So I'll just be moderating the chat to make sure that everything's safe. So you might be familiar with Zoom, you might not. So we've just got a couple of buttons here. So you can use the chat. You can either have that directly to panelists or myself. Oh, we've got another joining. Fantastic. Lovely. So yeah, you can put all your questions in the chat box if you'd like, or raise your hand as well at the end, just to put in your questions. So just a little bit about us. So I'm Cindy Smith. I'm a Science Engagement Officer for schools at the Wellcome Connecting Science, which is based at the Wellcome Genome Campus. And today, obviously, I'm joined by Matthew Colhill, who's a postdoctoral fellow, and he'll be talking to you later about cancer mutations. So if you wouldn't mind in the chat box adding what school you teach at, maybe the name and postcode is just for our records. So as I said, that we're at the Wellcome Genome Campus. So this is the Wellcome Genome Campus. It's really big. And on this campus, we are connected with Wellcome Sanger Institute, as well as MBIL EBI. So Wellcome Sanger Institute is mainly to do with wet lab kind of things, whereas MBIL EBI is more sort of bioinformatics and a lot of data storage. So these are some of the research that's being done on campus. So we've got human genetics, so the study of inheritance and how the genes are passed on. We've got cancer aging, somatic mutations, which is obviously the study of cancer. We've got cellular genetics, so the study of many different types of cells. We've got biodata, because obviously all of this creates so much data that we need to store. So we have a huge data center for that. We've also got the Darwin Tree of Life, which is sequencing every eukaryotic species in the UK, so over 70,000 species. And then we have parasites and microbes, a study of things like malaria and coronavirus, for instance. So we've been sequencing a lot of that. So we've also got live now genomics for educators. It went live a few weeks ago. But if you sign up before, I think it's the end of May, you have access for a whole year. Whereas if I think you sign up after May, then it might be restricted time-wise that you'd have to do it in 3 weeks. But if you sign up now, then you can do it, say, in the summer holidays when you might have a bit more time. So how the session is going to work today. So we've got lots more people joining us now. Fantastic. So how the session is just going to work today is we're going to hear from Matthew some more. Then he's going to answer some lovely questions that you might have regarding his career or the topic that he's going to talk about. Then I'll just give you some background into some teaching cancer mutations, resources that we can offer, and maybe get your input on any improvements that you'd like. So the session is being recorded and will be available on YouTube and its Learning Online portal. So now I will hand over to Matthew.
Okay, great. Well, thanks everyone for joining. It's a pleasure to talk to you this evening. Yeah, Cindy mentioned, please save lots of questions up and hope to have a lively discussion after the talk, and hopefully this is fun and interesting for everyone. So the talk will be about cancer gene editing and cancer mutations. And yeah, so my name's Matt Coelho. As Cindy mentioned, I'm a postdoctoral fellow at the Wellcome Sanger Institute in Cambridge. And yeah, a little bit about me by way of introduction. So yeah, I did sciences at GCSE and A-levels and maths, and then I went to university to study natural sciences here in Cambridge, and then specialised in biochemistry. And then I went to London. I did a PhD in cancer immunology and cancer signalling, in the Francis Crick Institute in London King's Cross. And then I came over back to Cambridge to first postdoc in AstraZeneca, so in industry, on gene editing, so CRISPR technologies, and then applied both of these fields together here currently at the Sanger Institute, where I'm applying gene editing to understand the function of mutations in cancer. So that's my story. So this is kind of a talk aimed at kids, which I kind of gave recently to school children, and hopefully we can open the discussion up, so you can use some of these materials for teaching, but hopefully you guys can also learn as well about what we do here at the Sanger Institute. So mutations, what are they and why are they important? And that's essentially the basic research question. So we study, sorry, I'm just moving the Zoom windows around a bit. Yeah, we're studying mutations in the lab using CRISPR gene editing. I'm sure many of you have heard of CRISPR. It's very exciting. We're changing the DNA. And we're using that to understand the importance of mutations and what they're doing to cancer. So for the first time, really, we're able to introduce mutations we find in the clinic engineer them into cancer cells to see exactly what they do. So we can analyze the function of these different mutations we're seeing. Okay. So step back, what is a mutation? Well, it's obviously a mistake in DNA molecule, which is the instruction manual of the cell, the hereditary molecule. And there's 3 billion of these bases that need to be faithfully replicated in each one of our cells every time a cell divides. And obviously that can lead to mistakes. And the mutation is essentially just a variant which is introduced perhaps by a random mistake in DNA replication, or perhaps by mutation from the environment, such as UV or carcinogens, for example. So these mistakes are often repaired by the DNA damage repair machinery, but the ones that aren't repaired can lead to certain diseases. Genetic diseases, and one of which is cancer, the main focus of the talk. So all of this information together, this instruction booklet, the DNA code, made up of A, T, C's and G's letters or bases, they all make up a genome. And this is essentially all of the genetic information in a cell. And each one of our cells has this identical genome, apart from the gametes, the sex cells, which are obviously haploid, so half of the material. And we can actually analyze or read it, read the genome, read these letters, like reading a book, using a technology called DNA sequencing. And the Sanger Institute is kind of a world leader in that technology. And indeed, the human genome was sequenced or read at the Wellcome Sanger Institute in Cambridge for the first time, which is a huge contribution, obviously for health and understanding diseases with a genetic basis, but also just enabling lots of other science. So the first kind of interactive question I have, which is kind of a fun fact, but I'm not sure, Cindy, if we have the interactive polls today, or if we're just going to run through. What do you reckon?
We don't have the polls today, so if everyone can just put the answers in the chat, which do you think it is? Okay,
great. So how many meters of DNA are there in each cell in our body? So 3 billion bases all squished together. Maybe people can have a quick go at that. Keep interactive.
So we've got 2 answers so far.
Is it A, 4 millimeters, B, 30 centimeters, or C, 2 meters?
I'll just give it a couple more minutes for anyone to see what they think.
Okay, great. Yeah.
Cool. So everyone's gone for C, 2 meters.
Yeah, excellent. That's very good. So yes, huge amount of material which is squished up and packaged beautifully in the form of chromatin in mammalian DNA. Proteinaceous complex keeps it together and packs it into the nucleus. So I think, yeah, one way you can think about it, although I can't really comprehend how far this is, but if you were to stretch out all of the DNA from all your cells, reach back and back to the moon and to the Earth 1,500 times. So it's a lot. Okay, so back to the mutations. This is essentially a substitution of G for an A. So one of these bases, and as I said, this can lead to some issues. So how do we actually analyze the mutation? So there is very little we know about what a single nucleotide change will do. So because if it's in a protein, it can make an amino acid substitution because, you know, the DNA is coding for a RNA and then that's making proteins. And then if that is a single amino acid change, that's actually is quite hard to predict if that's going to be detrimental for the protein, and then even further down the line, if it's going to actually impact a disease. For example, is it going to cause cancer? And if it's in a non-coding region, so if that mutation is in somewhere which doesn't code for protein, it's even more hard to predict what it's going to do because it may have an effect on gene expression, which is hard to define. So with these types of gene editing technologies, we can actually directly ask this question by making these mutations in the laboratory. So what can we do to DNA to analyze it? Well, one of the things we can do to this macromolecule is to sequence it, which I mentioned, which is reading it from 5' to 3' using the Illumina technologies, for example, which you've probably heard of. And secondly, we can do this kind of thing. So this is just a very simple gel, which is actually something I did in the lab a couple of months ago. And we can see here that this is a gel where we've loaded the DNA molecules on and separated them by size by running a current through the gel matrix. And so small molecules will go far further than large molecules. And you can see here that we have a nice band which we've visualized. So this is actually a visualization of DNA molecules by looking at them with UV and staining them with a certain molecule which intercalates with the DNA called ethidium bromide. So we can actually visualize DNA in the laboratory as well as sequence it. And this is what it looks like, as you know, high resolution, this model of the double helix. So this is where I do most of my molecular biology experiments. So I'm not sure how many of you have actually been into a laboratory, but you know, we essentially kind of mixing small volumes of liquid together in a very precise way, using these kind of pipettes, and these are the tips we use to move liquids from A to B. It's a little vortex to mix things. I don't think my bench normally looks this tidy, but it's for special photographic evidence. So hopefully that didn't bore you, because I'm not sure how many of you go into the laboratories on a regular basis. So back to mutations then, from the DNA to people, where mutations are manifested. And, you know, obviously we're interested in helping public health. So this is the ultimate drive behind this research questions. So with 8 billion people roughly on Earth, actually it's estimated that every tolerated mutation is present at least once somewhere in the human population, which is kind of really striking fact actually for me. So tolerated means that when a mutation occurs, the ones that are really detrimental will actually have a strong negative selection, like evolutionarily. So tolerated means that at least that the individual will reach sexual maturity and be able to pass on their DNA to the next generation. So we still don't really fully understand what all of these mutations do, and obviously we can't really sequence everyone. That would be the ideal. If we sequenced everyone's DNA, it'd be very, very expensive. So we're not really there. And find all these mutations and then associate them with diseases. Obviously, there's lots of ethical issues about what we do with that kind of data, which is a whole new discussion. But what we try and do is do that in the laboratory on cells. So we take out all of those factors and make it much cheaper and quicker. So back to so that's basically essentially what I just said. So with CRISPR gene editing, CRISPR-Cas9 gene editing, we actually make the mutations which we might see in a patient, and then directly ask the question in a cultured cell what that does outside the body. So it has some kind of caveats because this is all in vitro cell culture, we call it, in the dish. So does anyone have any questions so far? Just a very brief pause. Cindy, I'm not sure if you can direct the mic because I can't see the chat box. So.
No, that's fine. So if anyone has any questions, you can either unmute yourself or put it in the chat box. We'll give a minute for that.
Sure.
Oh, we've got someone who's going to ask. Are you unmuting?
Yes, my name is Clara. Hello.
Hi, Clara. Thank you.
Hi, Clara.
Yeah, first of all, thank you for doing this because it's really reassuring to have somebody backing us teaching this very difficult thing.
Mm-hmm.
And I am interested in knowing, that you sequence humans. I suppose you still do it live, right? I mean, you do it in the lab, but surely somebody must be still sequencing people around. Is that right?
Yeah.
Yeah, my question is, what happens if you do find a mutation that you are interested on or you're concerned about? Do you call the person? I don't know. What's the procedure usually? Thank you.
Yeah, thanks, Clara. Yeah, that's an interesting question. Yeah, so there's all sorts of ethics around how you set up a study, and they'll have very strict things about what the science is looking at. So at the Sanger Institute, generally, it's kind of not done concurrent with any clinical activity, if you like. So a lot of it will be done on healthy donors. And we're not running clinical trials, so it's not our kind of scope generally to interfere with clinical practice. Okay, when you're running a clinical trial, it's very different. That's something they'll do at Addenbrooke's, where they might sequence a patient biopsy, and that information is directly used to as a screening mechanism to influence clinical practice. Okay, so what we do is slightly detached from that, but ultimately we hope that our work will get into influencing clinical practice. But a lot of the stuff we do is not from healthy donors, for example.
Yeah, we do have Illumina on site, so they are more patient-based, and they get samples specifically from Addenbrooke's. But it's an all anonymized data, so it's the doctors that would send it. So if they've got something that they're nervous about, worried about, they would send it to, say, Illumina, very anonymously, so it'd just be a bunch of numbers. They would sequence it and send the data back after it's been analyzed, and then the doctors will know what they're looking at, like if it's a certain type of cancer, whether they need to approach it in a different way with medicine. But great question, Clara, thank you. So we'll continue with your chat.
Okay, yeah, thanks. Yeah, interesting question, so thank you very much. Okay, moving on. A little bit of an interactive one. Do you think that mutations are always a bad thing? Just a quick put it in the chat. A, yes, B, no, C, maybe. What do you guys think?
Yeah, so we've got a lot of B's. Yeah, the consensus seems to be B. No, or are mutations always bad? No.
Okay, yeah, well, I'll have to agree with you there. So there's always some genetic variation, and this kind of fuels evolution, obviously. So the selection for positive traits comes through the arising spontaneous mutations. So if we didn't have any genetic mutations, we probably would be evolutionarily millions of years behind where we are now. So there can be some beneficial mutations, although the majority are either neutral or deleterious, I'd say, so slightly rarer. That's the price we pay, and that's how life has evolved. So we have to deal with mutations, and many organisms have the repair machinery in DNA so that there's not too many mutations and it's not compromised really. So that's very good. So essentially, yeah, just these two discussion points. Why might mutations be problematic? Obviously, the hereditary diseases, so you can inherit predispositions to cancer, for example, but also yeah, they're beneficial for example, evolution, and several examples such as mutations in beta globin have been associated with resistance to malaria, for example. Some mutations, so they call sickle cell anemia. So it's a bad thing, but also, you know, you have some resistance to malaria, so that's beneficial. And another classical example is the cystic fibrosis mutation, which is obviously very detrimental, lung pathology, but actually these people are often very resistant to cholera toxin. So if there's cholera outbreaks, you're much less likely to be affected because of the mutations in this. So there's some reasoning why some of these problematic mutations have actually been selected for over time of human population on Earth. But generally we're focusing on the top question there, genetic diseases, especially cancer. So mutations accumulate over our lifetime, and the older we get, the more likely they are to accumulate enough mutations to cause cancer, for example. So how do we practically do this? So how do we make these mutations in the laboratory? Well, we generally grow cancer cell lines outside of the body, so human cancer cell lines. You may have read the book about Henrietta Lack, the HeLa cell lines, for example. And there's many hundreds, thousands now of these cancer cell lines which have been derived from a patient's tumor. And then maybe decades later, we're still working with them because they grow indefinitely on plastic. They're growing, the adherent ones, blood ones grow in suspension. And we add these medias to keep them healthy, which have growth factors, sugars, and things like that, building blocks of the cell. And then we grow them at 37 degrees in a humidified incubator, very sterile conditions. And then they all expand, and we can actually do some mutations on their DNA and see if they die or grow more, etc. So this is a sterile tissue culture hood, okay, which is what we've been doing today.
Just a quick question then, Matthew. The cell lines that you have, are they historic? Were they once upon a time donated by patients at the NHS, or how did you come by them?
Yeah, many of them are quite old, but they're all commercially available now. So the ones we work with are all commercially available, so you can buy them from authenticated providers, which means that there's strict ethics in place for these types of things. So the same is true, obviously, this is, and everywhere really, all research institutes that make sure that all the ethics in place that we can work with this human material, although it's, it's been, perhaps hasn't seen the human for many years. So there's no patient data kind of associated with these generally. It's all anonymized, as you say. That we are also deriving new cell models in, for example, the Garnet lab, which I work in. We also receive samples from hospital, for example, and then derive new cell models, but all the consent is in place for these types of procedures. So you have to agree to donate them to scientific research. That's a great question. Okay, so why are we interested in looking at understanding cancer genome function and cancer variant function or mutation? So as also mentioned by Clara, actually, this has a huge implication in the future for genetic testing. So if a patient comes in with a cancer, we generally would take a biopsy these days to test actually what genetic mutations are driving it. And that really transforms how the patients are treated. So the second point is the precision medicine. So that's what I mean by it directs how the patients are treated. So if you find one lung cancer with a certain mutation, you would treat it in a different way to another person's lung cancer, also lung cancer, but with another driving mutation, because we have medicines which are specific to those different pathways which are going wrong. So that's why finding out, what these mutations do is very important, because medicine A wouldn't work in patient B. So they're kind of personalized medicines, which is really improving the toxicity profiles for many people. Much better than chemotherapies, which is a kind of one size fits all and can have a bad toxicity profile. And number 3 is really understanding the disease itself. So understanding the fundamental biology behind it. And this really helps the drug development process. So we often collaborate with other pharmaceutical companies to accelerate cancer therapies for patients. And if we find, for example, mutations, so my boss was involved in figuring out how a certain gene goes wrong called BRAT, which is mutated in the vast majority of skin cancers. And now actually, years later, there's lots of different drugs against that protein which goes wrong in skin cancers. So by figuring out what that mutation did, actually directed therapies years later, which have been very useful. Okay, so I talked about how DNA mutations can occur through cell replication, and as we get older, these accumulate. So one of the logical steps forward is to think, well, perhaps large mammals such as elephants would get more cancer than humans. Do you think that's logical? So yes, if you think that elephants get more cancer than humans, or B, no. What do you all think? Just I'll give you a minute to put that in the chat.
Got a couple of answers so far, so we'll give it a couple more minutes. Someone says Peso's paradox.
Yes, okay, yeah, exactly. Yeah, very good. So you'll know the answer.
Yep, she said no. So we've got 1A. Oh, Pato. Is it Pato or Pesos?
Peto, I think, yeah.
Peto. Okay, so we've got mostly B's and 1A.
Okay,
yes.
Well, yeah, so they do not get more cancer than humans on average. And the reason that this is down to the control of DNA damage repair. So they may accumulate more mutations over their lifetime because they are quite long-lived and they do have a lot more cells than us, but they have this protein which we have a homologue of as well. TP53 is the gene, and it's actually kind of called the guardian of the genome because it really protects us from accumulating too many mutations. And it means that when a cell gets a DNA mutation, it's more likely to die than proliferate and carry on growing, which is really good because it's kind of a great cancer prevention mechanism. We've got 2 copies of this gene, like we do for most genes, 2 copies, one from mum, one from dad. But elephants actually have 20 copies. So they have huge abundance of this, which means that it's very unlikely that all 20 copies will go wrong. In cancer, the majority have some defects in this pathway. So actually, by chance, we have inactivation of both copies of p53 in most of our cancers in some cancer types, for example. So that means that if we had more copies, perhaps we would get fewer cancers, like elephants. Yeah, so that's just an interesting fact about the control of mutations and the tolerance of them. And this is excellent work from the Sanger Institute, which I wasn't a part of, but recently published in Nature from the cancer aging and somatic mutation groups in the Sanger Institute. But it's kind of reflecting on this idea of how DNA mutations may accumulate and then contribute to the aging process in all sorts of species and mammals shown here. So as you can see that as we age, in humans here, mutations accumulate. And so as we get older, it's actually more likely that we do get cancers. But it also means that this might contribute to the biological process of aging itself. But what was really interesting is that actually all of these species with different lifespans have very similar mutations at the end of their life. So there's kind of the hypothesis that perhaps the number of mutations may put kind of an upper limit on the lifespan for organisms, which is very fascinating. And as you expect then that the mouse would accumulate more mutations per year than the human. And that's indeed this correlation here between lifespan and the mutation rate. Okay, I hope that makes sense. So just to summarize what we've shown so far. So the DNA, the codes for all the genes making up our genome. This is the information booklet of the cell. And the mutations, these mistakes, can obviously be detrimental and cause many different genetic diseases, including cancer. And our job is to kind of figure out what they do by introducing those mutations into cells in the laboratory. Okay. So one thing that's really important in cancer is evolution on the cellular level. So I talked about how mutations can be beneficial for evolution at the species level, but this actually is fascinating when it kind of occurs on a cellular level as well. So if you think of the cells as individuals, they're all out to win, and they're all out to divide and beat their partners to the nutrients and growth factors in the environment. So actually this book, Origin, the Darwin's famous sketch, which was actually recently returned to Cambridge University Library. Actually, it's been stolen, I think, but safely returned, undamaged. This is the sketch of the tree of life, this speciation branching with different. This would be animals, really. So I think this. And indeed, this kind of evolutionary tree, where there's branching and selection, natural selection, occurs at a cellular level. And this is kind of an overview of what happens in cancer. So we think actually, for example, this can lead to drug resistance. So the natural selection would be to win out and be resistant to the drug, the chemotherapy, for example. So we have all sorts of mutations driving variation in the cellular level. We get a bottleneck here where we treat the patient with a therapy, and unfortunately we get the cells out which are most resistant to the cancer therapy, and then they will become the dominant species or clone in this case. So this is this kind of heterogeneity in cancer, which is been pioneered by someone at the Francis Crick Institute called Charles Wanton, who's very influential in this field, and it's kind of influencing how we think about treating cancers, because it's not just one, it's kind of a mixture of all different types of mutations that we're trying to treat, which makes it tricky, but it's transforming how we think about treatment. So a bit about my work in a bit more detail. So using CRISPR-Cas9 to make these mutations. So I'm sure you guys have heard this in the news, but it's like a molecular scissors which cuts the DNA, and we can kind of paste in the little bit of mutations, the mutations we find interesting, which might be from the clinic, might be found in a patient with cancer, or might be artificial completely. So yeah, so we then read out what happens by seeing if the cells die and sequencing the DNA. So this has only been made possible quite recently. There's huge amount of pace behind this research, and it's taken off obviously and transformed the whole world of molecular biology and beyond, and even therapies now for patients. But yeah, this was Nobel Prize in 2022 shared by Jennifer Doudna and Emmanuelle Charpentier on the discovery and implementation of CRISPR-Cas9. And they're involved in actually applying this for therapeutic applications, which are already being used for patients with cancer. We can discuss that at the end if you like, but also for diagnosis and what I do in the lab, so speeding up molecular biology and basic research. So this protein Cas9, which is shown here in a crystal structure, is in grey, and what it does is it sandwiches itself on the DNA and clamps it, and it finds exactly the piece of DNA out of all of those billions of bases by matching this guide RNA to a perfectly complementary region on the DNA. So, it has base pairing A with T, G with C, and it will find these 20 nucleotides which are exactly complementary by a two-dimensional scan. We're still figuring out how exactly that works. And then it will make a cut in the backbone of the DNA. So it's a double-strand break. And then that allows us to introduce mutations at exactly the programmed position we're interested in the whole genome, which is really transformative. Yeah, so sometimes this even Cas9, this search for the correct sequence can make mistakes, which means that it can lead to unwanted cuts in the genome that we don't know about, and also can be really can be an irritation if you're trying to implement this for a therapy where we really need to know what we're doing and be really safe and 100% on target, no off-target activity. So it does this because sometimes it has several mismatches and it still binds to the DNA, even though one or two of the nucleotides, the guide RNA, aren't a perfect match. So we call that an off-target site. So part of my work at AstraZeneca was to kind of stop that from happening and make this much more precise. And so I'm going to show you a little bit about that. Just on this very short video, which I'll talk you through. It doesn't have any sound, on this method called Crispr Guard, which we use to protect off-target sites, unwanted sites of cutting. So this is normally how it works. We can make our mutation here, but sometimes when there's a mismatch, it will still bind and obviously cause cuts elsewhere. So what we did was we added an inactive Cas9 for these off-target sites to block them and then protect them from cutting. And then only our on-target site is cut in this case. So that's how the technology works. And essentially it should improve the safety of gene editing, the predictability, but also we'll know better what we're doing in the laboratory in our experiments because it'll be much more precise. I hope that makes sense. So just to say, I don't work for AstraZeneca anymore, so that's all published. No disclaimers required. And just kind of wrapping up now. Yeah, so good times for questions. But just to summarize that, so the CRISPR-Cas9, we use that to make the mutations we're interested in and kind of cuts the DNA. We can paste in the mutations we're interested in a very simplified way. That's a bit of an oversimplification. but it does make mistakes. So we can actually use that mechanism to protect the off-target sites from cutting. And finally, yeah, we use these to kind of track this technology to track what the mutations do. So we have exactly a genotype to phenotype, we call it link. So the variant to the effect. And we're actually just making those links in the clinic and hoping they'll make a big translational impact eventually. So thanks everyone for listening. Hope you enjoyed that. It's a nice picture of the Sanger Institute. Hopefully we can do more in-person sessions soon. But yeah, I look forward to the discussion. Please fire away with any questions. More than happy to answer them for you. Thanks very much.
Fantastic. Thank you, Matthew. We've got one comment already from Clara in the chat saying we will reach the day when we can insert TP53 genes in our genome and no one gets cancer anymore.
Yeah, that's a really great comment. Yeah, and actually one of the kids has asked that recently as well, and I often think about that. So it's more of an ethical consideration than a scientific consideration, because yes, that is possible, and it probably would reduce cancer rates, almost certainly actually. But the ethical consideration is very important, because if you make a genetically engineered human, then any mistakes or unknown consequences will be hereditary, so they'll be passed down for generations and generations. So it's very risky, and obviously it's up for public debate, but there would be a lot of people against that for obvious reasons, because we're not fully aware of all of the unknown effects of these technologies. So we generally are using CRISPR-Cas9 editing as therapeutics in the clinic only for tissues which aren't mutations which aren't passed to the next generation. So in adults, where we'd take their cells out of the body, gene edit them, put them back, and then they're not passed on to the next generation, because we're not gene editing the germline. So sperm and eggs don't obviously ever see CRISPR-Cas9. Okay.
Fantastic. Thank you. Does anyone have any further questions for our lovely scientist, Matthew? If you'd like to unmute or put your questions in the chat. We're getting lots of thank yous for you as well.
Yeah, thank you.
So I've just got a question. What made you want to get into this kind of a career? What inspired you?
Yeah, thanks, Cindy. Yeah, so I think I always was interested in cell biology from university days, but had a few very inspiring lectures in university where I just saw how much of an impact that cancer research was making at that stage. It was a very exciting time, and I just got swept up in the excitement, because we're really well, I don't know, I'm super excited about this is the time to really be a scientist, because there's a revolution of CRISPR, we're transforming medicines. There's lots of exciting things happening all at once. And yeah, I think we've come a long way in cancer research. So immunotherapy was something I studied in my PhD, and it's actually completely transformed how we treat cancer patients across the board, basically, in the last 5-10 years. So yeah, I'm just super lucky and very interested, as well as thinking it's actually obviously a very good cause to get involved in.
What did you study for your current role?
Yeah, in university I did natural sciences and biochemistry. Yeah.
Very interesting topics.
Mm-hmm.
Lovely. Any further questions for Matthew? So we've got, do you consider epigenetic markers in your research?
Yeah, that's a fantastic question. Thank you. We do. Well, basically, yeah, that's a good question, but we don't actually generally know, because when let's say we have a mutation in a gene which encodes a protein, in the normal cell, it will be an A, and in the mutant cell, it will be a T. And if that changes an amino acid, we pretty much put all of the functional consequence down to that change in the protein. So the only difference between the cancer cell and the normal cell is that DNA change. So we don't think that single nucleotide change will really hugely impact the chromatin, the epigenetic expression or configuration of the chromatin. So yeah, because chromatin isn't very sequence dependent. It's more kind of context dependent, tissue dependent, etc. So just a single couple of mutations is unlikely to be driven through epigenetics. But yeah, that's a really good consideration.
Lovely, fantastic. Thank you, Matthew. That's a great question. If anyone has any further questions, we're happy to hopefully pass them on to Matthew, but we'll let you get on with your evening so you have the rest of your evening to yourself. So thank you so much for joining us this evening, Matthew.
It's been a pleasure. Thanks, everyone. Thanks, Cindy. Okay, take care. Bye.
Bye for now. Lovely. That was such a lovely talk from him. So now I'm just going to talk to you about some of the resources that we have, because some of you might have seen this already. Yeah, so I'm hoping everyone can see that in presentation mode. Yep. Okay. So some of you might already know about our yourgenome.org webpage that we've got. It's got a lot of resources that you can share with your students, activities that they can either do by themselves or activities that they can do with you. So one of them is some of the facts that we have. So you can search for different facts that some of our scientists have written, such as this one about what is a mutation. Then, just like Matthew mentioned earlier, we've got an activity on BRAT, which is about the gene to cancer therapy. Next slide. Real quick, there we go. So the BRAT challenge is learning the significance of that mutation in the malignant melanoma, just as Matthew mentioned earlier. So from some sequence sheets, they will need to find the BRAT mutation and identify which is healthy tissue and which is tumor. So you can print out several sheets that I think half of them are healthy and half of them are filled with tumors. So then once they find the right sequence to find which one has the BRAT mutation, they can put this into the Cosmic Online database to compare these sequences with those from cancer researchers. And this database, the same with everything that Wellcome Sanger Institute is about, is making sure that all data is available for everyone to have access to. So I'll just play this little clip. So this is one of the clips that we have on yourgenome.org, and it just shows you a bit more about gene to cancer therapy. So I'll just play a little bit of that when my mouse decides to find itself. Hopefully you can hear it.
Cancer is the change of what was once upon a time a normal cell into a cell which grows and proliferates in an uncontrolled fashion. Every normal cell has a blueprint, a DNA code that normally tells it how to behave. And if that DNA code or blueprint becomes changed or mutated, then that can lead to genes running amok and a cell growing in a completely controlled fashion. That makes sense. Malignant melanoma is a special type of skin cancer. We all have within our skin cells called melanocytes. These are cells that normally produce pigment or color. For example, when we put to the sun, sunlight is harmful to our skin. So normally these melanocytes will produce a brown pigment called tan, which helps protect us from this ultraviolet, this radiation. And unfortunately, if you're exposed to too much sunlight or ultraviolet radiation, then these melanocytes can accumulate mutations or damage in DNA. It's melanocyte, the normal melanocyte becomes a cancer cell.
So that's just a little clip of what we have on this page. And we have some, with this activity, some teacher notes, just to give you some overview of the activity and some background about it. But if there is anything that you think we should add that might be more relevant to the curriculum, or that you think might need a bit of updating, please do let us know. We're always welcome teachers' inputs. So another activity that we have on your genome is one called KRAS. And how these is to find out how these mutations alter the resulting protein produced. So it's specifically about the KRAS gene. In the classroom, you'll be able to model these proteins, so see how it is affected and why it is affected, why these proteins are affected. So we have some teacher notes on that one. We also have obviously all the worksheets that are easily printable. We also have a colorblind version as well, so it's friendly for everyone. But again, if there is anything that you think that we should add to that, that we'd always appreciate your input. And then just about the next talk we have coming up. So that is from a scientist called Megan Gossard, and she will be talking to you about T cells. I believe that's on the 8th of June, but we will be posting it on the It's Learning page, newsletter, and on our Eventbrite. So thank you everyone for joining. I will stop sharing. Find my mouse. Lovely. So we also have these genomics books, which was written by some of the staff that we've got on site, and it's got loads of stories about it, specific things, like, let's see, how did we get here, short history of pathogen genomics, those kind of things. So if anyone wants this book, we're happy to send them out to you. If you want to pop your address or just say yes in the chat, or if you want to message us directly, I will put our email in the chat. So are there any questions this evening?
Do you mind if I speak? Because by the time I type it, it will be ages. Do you do any school visits at all in your place? Or?
Yeah, so we are returning to a lot of campus visits. We're a bit fully booked for it this year, but for the next academic year, we are trying to book in a few more. So again, at that same email address, if you put in your request, if you want to come and visit the campus, and one of the team will get back to you. Fantastic. So thank you everyone for joining us this evening. I see we finished quite a bit earlier, but that'll give you your evening back. And I hope you have a lovely evening and hope to see you all in the next one.